Welcome, first years! Today's topic is tertiary protein structure, a crucial concept in biochemistry. A molecule's structure directly affects its function, and even a small change can lead to big functional problems. Scary, but let's dive into it. In order for us to be guided by our lesson topic for today, here are our learning outcomes. By the end of the lesson, I can be able to analyze the different stabilizing actions and the importance of these interactions to the other structures. Be able to identify and differentiate the tertiary protein structure from the different structure. Be able to understand the process that the tertiary protein structure does in order to define its essentiality as well as the consequences that it brings if misfolding happens. Next is understand the importance of the tertiary protein structure in our body's biological function. And lastly, be able to perform the post-discussion activity to further assess understanding about the topic. Now, let us start.
A tertiary protein structure is a 3D shape that a polypeptide shapes into. It involves twists, bends, and bonds that help it function and activate. It plays a key role in understanding its patterns, concepts, and overall the biological importance of protein in nature. Before we proceed with the complex part of this lesson, let's first get to know the tertiary structure. What makes this different from the other structures, and how does this folding happen? Tertiary structure is the 3D shape of a single polypeptide chain, formed as it folds upon itself, bringing distant amino acids closer together. This precise folding is vital for the protein's overall function.
Proteins fold into different patterns: globular like enzymes or fibrous like collagen. If the tertiary structure is disrupted by heat, pH, or chemicals, the protein becomes denatured and loses its function.
A lot of information, right? But now, let's head on to an overview of the different foldings under this structure. Proteins fold into a unique 3D shape using secondary structures, forming their tertiary structure needed for proper function.
Alpha helix for flexibility and beta sheets for strength, these are two common foldings that are stabilized by hydrogen bonds. In a tertiary structure, they fold into a compact 3D shape that forms functional sites that are vital for a protein's role. Protein folding is guided by stabilizing forces, not random. These forces help proteins fold precisely, ensuring that they can carry out their specific functions effectively. First, we have hydrophobic interactions. These involve nonpolar side chains that avoid water by folding inside of the protein. This helps the protein become more compact and stable in its 3D structure. Next is hydrogen bonds. These occur between polar side chains or between a side chain and the backbone. They help stabilize the folds by forming weak but important links.
Now, let us again recap and briefly discuss the two mentioned forces, starting again with the hydrophobic interactions. These interactions again fold inside the protein, and this interaction is one of the relatively stronger interactions among the others, such as your hydrogen bonds. Here are the following strengths of the hydrophobic interactions. First is temperature. Now, as your temperature increases, the strength of your hydrophobic interactions increases as well. However, take note that at an extreme temperature, hydrophobic interactions will denature. Next is the number of carbons on the hydrophobes. Molecules with the greatest number of carbons will have the strongest hydrophobic interactions. And lastly, we have the shape of the hydrophobes. Aliphatic organic molecules have stronger interactions than your aromatic compounds. Now, the branches on the carbon chain will reduce the hydrophobic effect of that molecule, and linear carbon chain can produce the largest hydrophobic interaction. This is because carbon branches produce steric hindrance, so it is harder for two hydrophobes to have very close interactions with each other to minimize their contact to water. Our next interaction is the ionic bond or salt bridges. Now these are formed between your positively and negatively charged side chains such as your lysine and your glutamate. Now they act like magnets as they keep the protein structure locked in place. And lastly, we have the disulfide bridges. These are strong covalent bonds between the sulfur atoms of two cysteine amino acids. Disulfide bridges act like staples, holding the protein shape firmly in place. Let us once again recap or recall the last two forces before we sum it up to make sure that you have prepared yourselves as we will have our assessment later. Again, the ionic bonds or salt bridges are formed between two opposing charges. Ionic bonds result from electrostatic attractions between positively and negatively charged side chains of amino acids. For example, the mutual attraction between an aspartic acid, carboxylate ion, and a lysine ammonium ion helps to maintain a particular folded area of a protein. Now, let us head over to the disulfide bridges. As for this kind of interaction, two cysteine amino acid units may be brought close together as the protein molecule folds. Subsequent oxidation and linkage of the sulfur atoms in the highly reactive sulfhydryl or your SH groups, known as also your thiol groups, leads to the formation of cysteine. In some proteins, disulfide bridges are crucial and essential for their function. For example, in antibodies, disulfide bonds contribute to the stability of the antibody structure. They also stabilize the binding site for the antigen, which is responsible for recognizing and binding to a specific target. Enzymes also rely on disulfide bridges for their catalytic activity by stabilizing the active site of the protein. Well, isn't it fun to understand these concepts? But let me ask you, are you still there? Because here is a better view of how these interactions are found when plotted in a tertiary protein structure. Isn't it fun to learn and understand why these forces are crucial for our biological functioning, as it helps in keeping the folding stable? But not only that, because at the same time, it keeps us safe from misfolding. Well, misfoldings are scary. But here, we have our hydrophobic interactions, hydrogen bonding, ionic bonds, and last but not the least, our disulfide bridges. I'll give you a minute to familiarize yourself with the photo provided. Time's up! Now that you already had time to familiarize, we will have a short assessment. Label down all the patches with the one same as the previous photo, including its chain structure and the type of interactions. Do not worry, as this will serve only as your exercise. If you have any questions, feel free to comment down. For this activity, again 1 minute will be provided for you to answer the following. The time starts now. Time's up! Now, check your paper if you got the correct answer. Well, at this point, we hope that you have learned something based from our discussion. Now, we will summarize the tertiary structure of proteins. This is the interactions between amino acid residues side chains which forms the 3D structure of your protein. The tertiary structure is where the protein begins to take on its final three-dimensional shape via polypeptide side chain interactions as opposed to primary and secondary structure wherein the amino and carboxyl groups create the bonds. Tertiary structure interactions are not limited to localized regions of the polypeptide chain. Amino acids on opposite ends of the chain may interact, unlike in primary and secondary structure where residues in close proximity interact.
Now that we have explored the different forces that stabilizes the tertiary structure of a protein, we can say that the folding of a protein is very delicate and precise. Now, let's dive on what will happen if a misfolding does happen. When a protein misfolds, it loses its function because its shape determines how it binds or interacts with other molecules. So, if the shape is wrong, it can't or won't work properly. Misfolded proteins can also form toxic clumps or aggregates that build up in cells, causing damage and leading to diseases like Alzheimer's, Parkinson's, and Huntington's.
So now we know that protein misfolding is not a small problem we can ignore. These can actually lead to major problems in the body leading to diseases. Now let's do a quick recap. Tertiary protein structure is the 3D shape a protein folds into, essential for its function. It's stabilized by hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bonds. Key patterns like alpha helices and beta sheets help form the shape. When proteins misfold, they can't function properly and may cause diseases like Alzheimer's, Parkinson's, and Huntington's.
Before we let you go, of course, we have to make sure that you have understood the topic. Right, Gim Gim? Now, we will have a short assessment for only a minute and answer the following questions as fast as possible. Number 1. Which structure is directly affected when a misfold of protein happens? Number 2. What bond is the strongest stabilizing force in the tertiary structure? Number 3. A protein structure that includes alpha helices and beta sheets. And last but not the least, what covalent bond is formed between two cysteine residues? Now, let's check if your answers are correct. Here are the following answers. Did you get it right? Well, we hope so.
Now let us see if we were able to do the following. We did analyze the different stabilizing actions. We also did identify and differentiated the tertiary protein structures from the other structure. We also did able to understand the process that this structure does as well as the consequences that it brings if ever it misfolds. We are also able to understand its importance as well as how it really functions in our body. And for the last part, we will just provide a link in the description box for you to answer to serve as your review for today's assessment. The tertiary structure of proteins is more than just a scientific concept because in biochemistry, it's not just what something is, it's how it's built that truly defines what it can do. Now, see you soon!